Okay, T cell. Again, what does the red triangle indicate or represent? Antigen. In the case of the B cell, we said the B cell can be directly activated by the antigen. That is not the case for a T cell. The T cell has to go through a process called antigen presentation. And based on your knowledge of English, what does that mean? Has to be presented to it, exactly. So thus comes our friendly dendritic, or this cell. This called the antigen presenting cell, APC. And there are several types of antigen presenting cells. You have a dendritic cell. You have a B cell. You have a macrophage. Those are the three main types of antigen presenting cells, which means these are the only ones which can present the antigen to the T cell to activate it. So this dendritic cell engulfs the antigen and will then present it on the surface. Remember talking about the fluid mosaic model in NP101? This fluid can change. Well, one thing it does, you can actually get the antigen and put it onto the surface of the membrane. Now if you look, you've got two proteins, two particles sticking out of the membrane. You have the antigen sticking out, and you have another particle sticking out. Called MHC2, which is major histocompatibility complex class 2. Every cell in your body has an MHC. Now most cells have MHC class 1. These dendritic cells have MHC class 2, but all the other cells have MHC class 1. The significance of that, remember we said in the non-specific, in the natural killer cells, Monday, if they don't have the MHC1 marker, it'll destroy the cell. All cells have the MHC marker. The dendritic cells have MHC class 2, which identifies them as a specific type of cell, which means it allows this cell to present the antigen to the T cell. That's why this class 2 is so important. Class 1 cell can't present it. It has to be a class 2 cell. So if you look, as you can see, the antigen will fit in the receptor site for the antigen. At the same time, the MHC marker will fit in the receptor site for the MHC marker. Now there are actually two main classes of T cells. CD4 and CD8. CD stands for cell differentiation. CD4 is normally a helper cell. CD8 is normally your killer cell. So in the T cell here, it should really be either a CD4 cell or a CD8 cell. I'm just using one cell to show a point. So given we have that specificity we discussed earlier, now the T cell has been activated, and now it'll start to mature and divide. Remember we said the immune system has to have a memory, amnestic response. So here you go. You have the memory T cells produced. So the second time you're exposed to the disease, how many T cells do you have, relatively speaking, the second time? A lot more. You have regulatory T cells. We don't want this process to get overwhelmed because you make too many T cells. Ever heard of leukemia? You don't have too many T cells. So this helps regulate or back-regulate or negative homeostasis to maintain control of this, doesn't run away. The fun one is your killer T cell. This is the one that does the damage. And there's three main ways this cytotoxic killer T cell works. One method is it secretes tumor necrosis factor. Rapidly growing cells, those affected with viruses or tumors, will go through what's called cell death or self-cell death. They'll kill themselves. So the cells, they sacrifice themselves, but they kill the pathogen at the same time. Another way they kill is through interferon. Briefly, who remembers how interferon works? Okay, Tam's going to tell us. How does interferon work?
It interferes with it. It
How?
interferes with the division.
How?
The dying cell secretes to the adjacent cell so that it has like an immunity to.
Right, so the cell gets affected with virus, it secretes interferon. What effect does the interferon have on the nearby cells? What does make those cells produce? Remember there's antiviral protein to make? What's the name of the antiviral protein? What's the name of the antiviral protein that the interferon makes the cells make? Antiviral protein. The interferon makes the cells make antiviral protein. That's what prevents the growth of bacteria, of the virus. Third is perforin again. And then you got the helper T cells. The helper T cells do just the help with memory cells, regulatory cells, help killer T cells. They also help, let's roll over here just a little bit. With the B cells. Remember I said earlier that the B cells have to become activated or have to be put in overdrive. That T cell does that. That comes the same helper T cell here. Now here's where HIV comes in. If this helper T cell is dead, are no longer effective, no longer functional, will there be any cytokines to support the B cells? Therefore, will the B cell work? They don't work, will the T cells work? What happens to the immune system? Stop working. Do people, will a patient die? A very precise question here. Will a patient die of HIV? No, they will not, because HIV is not fatal. What does the person that has HIV succumb to?
Other disease.
Any opportunistic infection, exactly. Pneumonia, fungal infection, whatever it happens to be, that's what kills them. So that's your link then between your T cell and your B cell side of the house. Also, one of the differences we said earlier is that a B cell can be activated by direct contact. A T cell has to have what? Antigen presenting cell. Remember that. When you do all in clinical immunology or pharmacology, you need to remember that. But remember all of it, that's why I'm teaching it. Now, in regards to the activation, I'm going to do kind of a side step here before I run out of time. And this may help you understand why the lymph nodes are built like they are. Where are the B cells located? On the cortex or the medulla? Where are the T cells located? Based on what you just learned, why are they positioned that way?
Lymph goes through the B cells first and it does its
Right.
activation.
These become activated first, act as antigen-presenting cells, then activate the T cells.
Well, because they have to be presented to, right?
Yeah, not the B cells. The T cells have to have it presented.
Right.
That's why you don't have this process. You don't have the T cells outside and B cells inside. Because these will be the first to get the antigen, but can T cells be activated by direct contact with antigen?
No.
No, they must be presented. So you have to have the B cells first, they become activated, and they can actually now activate that T cell. Then, while later you activate the T cells and your antibodies. Okay, question about the T cell branch of the adaptive immune system. Okay, again, that should